What if the next medical breakthrough could change thousands of lives? Welcome to Moving Medicine Forward, the podcast that pulls back the curtain on the breakthroughs shaping the future of healthcare. I'm your host, Jeremy Schran, and together with CTI's leading experts, we explore the cutting edge of clinical research transforming patient care. Each episode, we dive into the science, strategy, and innovation driving medical advancements, joined by global therapeutic experts who are redefining how we understand and treat critical disease. From rare genetic conditions to revolutionary treatment protocols, we're not just discussing research, we're sharing the human stories that move medicine forward. Today we are joined by Dr. Robert Fisher, one of the true pioneers in transplant surgery, with a career spanning decades at the forefront of innovation in organ transplantation and emerging cell and gene therapies. In this episode, we explore the rapidly evolving field of xenotransplantation and why this moment may represent a real turning point. Dr. Fisher shares firsthand insight into the breakthroughs making this possible, from CRISPR-driven genetic engineering to advances in immunosuppression and clinical research infrastructure. We'll also unpack what this means for patients, the future of transplant access, and how disciplined, collaborative clinical research is helping translate groundbreaking science into real-world care. Welcome to the podcast, Dr. Fisher. We are so excited to have you on today.
Well, thank you, Jeremy. I'm always happy to be anywhere that I can aid patients and CTI's progress.
So let's start off. Can you walk us through your long career and the path into transplantation and what initially drew you to this field?
Quite honestly, I was always drawn to the field of transplantation and more importantly to surgery, even when I was still in college before going to medical school. I was very fortunate because I trained at Baylor College of Medicine with pioneers like Dr. DeBakey and Stanley Crawford. And of course, many of the innovations in vascular surgery and in transplant were developed with those initial parts of my medical school training. And then really, I think it was the bubble baby that I helped take care of with Ralph Feigen at Texas Children's Hospital, where it seemed so important that we needed to be involved in not only whole organ transplant, but in cellular and genetic transplant, now called gene therapy. So I think those were my earliest. And then I was fortunate as a surgical resident in my first year at Case Western Reserve, maybe it was the second year, to scrub with Tom Starzl on the first multi-organ rapid retrieval of all organs in a brain-dead organ donor. That was published in SGNO back in the time, 1982. And at that point, I was hooked that the only form of surgery and medicine I wanted to practice was transplant in all ages.
You've seen transplantation evolve dramatically over the course of your career. Given the long history of challenges in xenotransplantation, what recent advancements make you believe this moment represents a real shift in the field?
Yeah, my involvement goes back to really the 60s and 70s, and then working with the pioneers that did early xeno, Dr. David Hume's team at the Medical College of Virginia. H.M. Lee was actually my chairman and boss in 1991. And I think having been through the 1980s with the starts and stops with some of the pioneering work by Dr. Platt at Duke and David Sachs, who really has been working on his pig model that now the organs are being used for transplant. I really felt that the changes came when CRISPR-Cas really became a way that could rapidly and significantly improve genetic manipulation of cells and even people and animals. And I think that also was combined with another great development was the fact that co-stimulation blockade had finally jumped from mice to men and was going to be feasible and actually the most important way to maintain xeno organs in humans. And so, again, those two very important contributions colliding at the same time make me feel that these early clinical studies now that CTI is involved in, kidney and liver, are going to proceed to fairly rapid, larger clinical trials in the next year or two.
It's truly amazing to see this science moving forward.
Absolutely.
Speaking of breakthrough science, it's only impactful if it can be translated safely. What recent advancements have been most critical in making a xenotransplant viable, and how important is disciplined clinical research in that process?
What we've been able to do is been based on the shoulders of so many men and women that have dedicated their whole careers and lives, really since the 1950s, 1960s, to make this possible. And again, I think now, because we have at least three very strong bio groups that have strong enough funding and have developed literally pigs, one by natural selection that Dr. David Sachs' group has developed, that have almost identical swine leukocyte antigens. Every one of them can be brought up to large enough volumes to treat hundreds to thousands of potential patients. So it's scalable now, and even better than human transplant, where we can't say that all the HLA antigens are 95 to 96% identical. We can with these pig models. And now we also have one group that has been developed by repeated recombination changes with natural selection that no longer produce the main porcine antigens that all of us, including primates, all mammals, react to. The alpha-gal is called, and in fact, this is very important in preventing the acute loss of the organ by it clotting because of these antibodies. So that's a key factor. And then these organs, like two other groups that have been able to be developed by very important development of being able to take the genetic material, add or subtract particular genes, for example, remove all of the potential particles that would create these porcine viruses or xenoviruses to affect mankind, and be able to remove them by this and then re-inject those genes, that DNA, back into the cells and then implant them into a pig uterus and allow those now constructed identical piglets to develop. Also, the fact that the organs have also been developed in a series of either genetic or rebreeding that allows the organs to be the right size and not continuously outgrow the size of the human being that they're in. What we forget is that pigs have been engineered to produce food for humans. So we wanted them bigger with more meat, right? Now we really want pigs whose organs match, not just anatomically, but match for size.
It's very interesting. So, these xenotransplantation procedures have drawn widespread attention. From a clinical researcher lens, what should people understand and be cautious about when interpreting these early milestones?
I think that what they should be not cautious about, but they should be reminded that this is not science fiction. This is not creating a Frankenstein. This is actually using the right animal model, because unlike primates, where there was never a way to scale that, and also a question about the true ethics of doing that, there is no ethical boundaries about using pigs, especially pigs that are cloned and engineered for this. And also the fact that if we know now how to remove those potential viruses that were always a black box, and we now have shown, especially in the porcine islet studies, that they never created a clinical problem over years in humans. And now the additional safety factors that are built into these clinical trials, that is not something to be feared about. And I think the way that the centers that have transplanted the initial 9 patients have gone about educating and choosing those individuals that were very carefully educated, even to the point where there are some nice reviews of what those patients at, let's say, the Mass General and NYU had to convince their families about. And so this very important education that goes in to make sure that we're handling all the ethics.
So let's kind of dive into the difference. Like you mentioned, it's not Frankenstein. It's not science fiction. It's science. But it is taking a pig organ and putting it into a human. What are the differences, and what do these differences that exist mean for a patient and organ monitoring, safety considerations, and like day to day life for a transplant recipient?
The wonderful team and the wonderful patient at the Mass General and also at NYU over a 9-month period have demonstrated it. So some of the early concerns that we have and we still have with the ongoing clinical trials that we're participating in are with the fact that certain hormonal productions by the kidney are different and not as effective as in the human. For example, the hormones that are produced to help prevent anemia and to promote red blood cell is less effective in the pig kidney. But with the human kidneys that are not working still in place, they're producing some of that hormone. And the hormones called renin-angiotensin aldosterone that affect how different salts and water is either reabsorbed or excreted is different and will need to be carefully managed in each individual patient. And this isn't anything new. We saw this with the early really done back in the 60s with the use of chimpanzee organs and the volumes of urine that they might produce that led to difficulties in just keeping up with that. So again, there are differences, but more importantly, the anatomy of the kidney, the ability to actually have it function immediately and not turn black because of these preformed antibodies is now been eliminated. And we know that the ability for these kidneys to provide functional survival, lower the creatinine, and provide a what we call a GFR, a glomerular filtration rate that will sustain life, is being done in the models that have already been performed.
So you mentioned earlier that it was scalable. So what are the key logistical considerations in managing organ sourcing, harvesting, and planning the transplant surgery? And how can CROs support these highly coordinated processes?
Those of us like myself that have spent 36 years and organ preservation and transplantation. I've myself have procured thousands of organs and then transplanted them. And understanding what the issues are related to cold ischemia time, warm time. Are there certain factors related to preservation that are different in these organs? There has to be the logistics of making sure that the procurement and all the necessary data that has to be acquired, that this is the right organ for that particular recipient, that all of the infectious diseases, all the potential zoonosis, so xeno-related infections can be handled or be prevented for this particular recipient. And then all of the needed informed consent. For example, the caregivers that are going to work with these patients and with these organs about what must they be willing to submit themselves to be under constant vigilance so that they don't become a potential for carrying a xeno infection or another form of a protein that we just don't know about. So again, and the CRO has to be intimately involved in working with the group that is developing these herds of animals and making sure that they're produced in a both a ethical as well as a repeatable and verifiable manner. And then the fact that the organ that's being transported, where the team that's receiving that can have everything that they need to make sure that they safely, with all safety indices, protect the patient.
It's so fascinating to me, like all the logistics that go behind it and all the things that the science has discovered over the years to make this even possible.
Well, in logistics is another area that, the patients, the recipients never hear about, right? Um, what did it take for the teams to procure those, even the human organs, and what needed to be done to transport now that organ from, let's say, Los Angeles to Covington, Kentucky, or Cincinnati, Ohio? Again, all of those are part of the way that we make this work and effective and able to be repeated with excellence, not hit or miss, but with excellence and perfect coordination.
When you think about immunosuppression in xenotransplantation, what approaches appear most promising today and why?
Well, I'm also again feel that the important work that was done by really David Cooper and Kobayashi and the group at Maryland and at the Mass General, and even the very important work that was done by now some of the leaders in transplant at Duke that showed that calcineurin inhibitors, the standard FK506 and cyclosporin, were not going to be the drug that was going to prevent in general, if you can get past the initial kidney from being lost by the antibodies that are pre-existing, now you can get past that. The problem in xenotransplant is more related to antibody-mediated rejection. And so calcineurin inhibitors aren't very good at preventing that at all. So the development of the pig kidney transplants that were making it out beyond a year in primates using a co-stimulation blockade product that is now being used in humans and being tested and safe, it was another one of the primary forces that is making this, pushing this to possibility. And so, yeah, so that's the immunosuppression that will be used, and for the most part, the most important was what's called the CD40, CD154 co-stimulation blockade drugs that have made the pig organ to primate transplant successful, and are now obviously in phase 2 trials in humans.
So, xenotransplantation brings unique immunologic, infectious, and ethical considerations. How must sponsors, investigators, and research organizations work together to manage those risks and responsibilities?
I think the best way is by how we educate one another. And for example, probably the biggest force that helped the FDA decide that it was time to allow pig heart to be used compassionately in a human in Maryland was the National Kidney Foundation. Patients and their donors pushing to say, look, we know that the number of transplants has increased, but it's not enough. We're talking about 1 million or more patients with renal failure that never will be even listed. We're still seeing the fact that we have people that are waiting up to 5 years to be able to be transplanted. And if you're 60 or over, the chances that you will die, half will die before they get to that point is the reality. So again, what we need to do is we need to be very, very open, and everything needs to be an open book. These are the risks. This is what we don't know. This is what we do know. And for example, the initial using brain dead organ donors who couldn't give their organs, but now became the recipients of these initial pig kidneys. The fact that for example, Alabama, Birmingham's group and Jamie Law were very concerned about how long should we keep a family who's willing to give their loved one as a donor for this type of experiment to not end the experiment. Is it appropriate, let's say if the kidney keeps working for more than a week, is it really appropriate to ask a family to not end their grieving and by a week? What is rational and what is ethical? And I think if we have those of us asking those questions that are pushing this forward, that reassures the society that is going to gain the most from this, that we really have their benefit in our best interest.
If xenotransplantation ultimately becomes clinically viable, how could it reshape transplant waitlists, patients' access, and then long-term follow-up models?
It's going to be transformational in that we're talking about the hundreds of thousands of potential transplant recipients right now that die every year and every day because they can't get an organ. And then what about the retransplants? And what about, we're only talking about a third of the population that ever with renal failure or with organ failure that ever makes it to the waiting list. Now think about if the paradigm changes, that someone doesn't have to die for another person to live, because now the organs can be tailored and ready for when that individual needs it. And now, instead of developing allocation protocols based on how sick you have to be before you get that first access to the organ, now changes to how much better earlier can we transplant you so that we prevent many of these complications that prevent that organ from keeping you alive for 30 or 40 years fails because we let you get too sick and other systems are failing before you're allowed to get that chance for that organ transplant. So it's going to change everything.
It's some exciting stuff. Very exciting stuff. So over the next 5 to 10 years, what milestones should signal that xenotransplantation and the clinical research that supports it is ready for broader adoption?
I hope it's not going to be the 10 years that I spent pushing for liver transplantation to become a Medicare-approved procedure. So I think what will tell us is when you can get a kidney transplant from a pig organ and it is actually accepted and paid for by your insurer or by Medicare or Medicaid, then guess what? Now the standard of care.
Thank you so much, Dr. Fisher, for joining us today. I'm constantly reminded of all the brilliant people that I get to work with day in and day out, and you are absolutely no exception. It's really great to hear from you, and this is really exciting stuff and really future of medicine and how we continue to move medicine forward.
It's an absolute privilege and an honor for me to even be here today and also to be a part of this area of medicine, science, and surgery. So thank you.
That was Dr. Robert Fisher, offering a compelling look at how innovation and clinical research are coming together to reshape medicine. As xenotransplantation continues to advance, the potential to expand access and save more lives is truly transformative, and it's work CTI is proud to be a part of. And as always, thank you for listening. If you enjoyed this episode, be sure to subscribe, share it with a friend, and leave us a review. Visit ctifacts.com to learn more about how we can help you with your clinical trials. Until next time, stay curious, stay compassionate, and keep moving medicine forward.